Hi everyone, welcome to explaining concepts about biology, and this will be my presentation for Biology 110. So first, I want to tell a short story before we start this presentation. So actually, my dad, he got colon cancer one summer, and I vividly remember this because I sat in the hospital multiple nights. To be honest, when he first got his diagnosis, he was slightly scared, but good thing it was only stage 1, colon cancer. Cancer can be a frightening word as per the story that I just told. So we are going to be talking about cancer cells. And here is the overall agenda. So we will go over how cancer occurs, mitosis, a lab regarding mitosis and comparing cells, the cancer cells versus normal cells, and if there's actually a cure for cancer. And we also go over an example experimental treatment regarding colon cancer, since I just told a personal story about that. So, how does cancer actually occur? So, let me first explain the process of mitosis. Mitosis is a process in where the cell divides into two identical daughter cells, and this is important for replacing cells that are damaged or for cell growth. And also, and also, it is divided into G1 S phase, G2 phase, and these make up a stage called interphase. And mitosis itself is the actual dividing process, and cytokinesis is comes after. And so through this mitosis and interphase, there are actual checkpoints that the cell needs to be aware of, such as in G1, there is a checkpoint to check if the cell grows well enough and if the DNA is damaged. And in G2, it checks if the DNA replication is correct, like in S phase, and if it is growing well enough, and if the cell has everything it needs. And the last checkpoint is in metaphase, which is the fourth stage of mitosis. So there are these stages of mitosis: interphase, where there's the duplicate chromatin, the prophase, which is the early mitotic spindles start to form, and the prometaphase, which the spindles start to attach to the chromosomes, which are made up of DNA, and metaphase, which they line up, getting preparing to divide like an anaphase and telophase. You can see the cells start to develop into two individual cells, and then cytokinesis, they finally split apart. But mostly the checkpoint is in metaphase, where it checks to see if the spindles are correctly aligned up and all attached to the spindle. But what happens? But if this is all good, then the cell can go ahead to divide. But what if the cell is not is what if during these checkpoints the cell cannot go on? Well, usually the cell will it will happen in two different circumstances will happen. First, uh, first, it will go into cell limbo, where cells cannot continue to divide anymore, or it will go to apoptosis, which means that the cell will be directly killed off. But cancer is when there's the absence of apoptosis, where the cell continually rapidly multiplies without any checkpoints or control. So here is the lab section of our presentation and how mitosis can be viewed in real life. So the objectives to this lab, this is from lab 4, to view mitosis under a microscope and gain newfound understandings of the process. Here are the materials required, such as hyaluronic acid, garlic root tips, which can be swapped for other cell types, such as animal cells. In this case, cancer happens in humans, so animal cells will be closer to the structure of human cells. And the fixative solution, which is ethanol and acetone in a 3:1 ratio, microscope slides, cover slips, scalpel, razor, or scissors, forceps, pencils to apply pressure on the cover slip during the squashing the sample that you just made, and watch glasses to hold the root tips during the stages of treatment and staining. And finally, we have our stain, which is 8.8% Genesian violet. Excuse me. And then we have the mitosis um, steps, lab material steps. First is cutting the root tips. You need at least 4 to 5 root tips in case of mistakes. And fixing the root tips, so you need to put them in a glass container with the fixative solution that I just mentioned before, and ensure the root tips are fully submerged. Let them sit in a fixative solution for 3 minutes to dehydrate the cells and preserve the chromosomes. And then we need to soften the cell walls and transfer the root tips into a watch glass and add a few drops of hydrochloric acid and leave the root tips for 5 minutes. Make sure your skin does not come in contact with the acid because it is toxic. And after 5 minutes, gently wash the root tips with water using a bottle or dropper 2 to 3 times. So here are the last couple steps in this lab. Uh, this is staining the root tips. So we have to transfer the root tips to a clean watch glass and add a few drops of the Genesian violet stain and let the root tips soak for 3 minutes. So after those 3 minutes are up, wash the root tips in water 2 to 3 times and remove the excess stain, and then blot off any excess water. And to prepare the slide for the last step is to place this stained root tip on a clean glass slide with a drop of distilled water and carefully position the cover slip over the root tip. And make sure one edge touches the slide to prevent air bubbles and gently lower the cover slip. Apply light pressure using the eraser at the end of the pencil to squash the tissue and be careful not to break the cover slip. And then you have your slide sample ready for this lab. And then you can begin viewing the slide under the microscope. Why why is this lab important uh is because you can view these cells and gain understanding to how this process works. And scientists also use similar observation under the microscope to determine if they are cancerous or not. So this is what normal plant cells look like. This is from the lab, and you can see the different stages. As a reminder, here are the different stages, and they all have the shape and structure that is uh, appropriate for this stage. And here are the stages more closely in a detailed sketch. As you can see, uh, it's metaphase, also metaphase, and anaphase, etc. But cancer cells are way different because since they are rapidly dividing and they often have mistakes within the structure of their cells, such as they have a small cytoplasm and they can have multiple nuclei. And also, they also do not retain their shape. For example, this is the colon cancer on the right, and it has a strange shape compared to the regular cells around it. They often also bunch together in a strange way, also like this. So what are the other factors that cause cancer? Other factors include lifestyle and diet and genetic factors as well, because there are genes for cancer that can be inherited. But for lifestyle and diet, let me give some examples. So here's a chart for the lifestyle and diet that can possibly contribute to higher increase of cancer risk. So in the yellow, we have, possible suggest but limited suggested risk for cancer if you do these things. And the orange, it gets worse. So you have a higher chance of getting cancer if you do these things. And the red is the very, it is very bad. It is the worst possible thing that you can do to have a very convincing risk for increased cancer. So, what is the specific cure for cancer? Actually, there's no specific cure, but however, there are many treatments, such as chemotherapy, which is a type of drug treatment that kills cancer cells and controls their growth or relieves disease-related problems. It may involve a single drug or a combination of two or more drugs, depending on the type of cancer and how fast it is growing. This these are, for example, you can also get it injected, and there's also different types, such as radiation treatment. There are also more types than I just mentioned, besides the uh, besides injecting into the arm and radiation, but I won't go into that. You can look it up on the internet if you are really curious about those different types. So here is an example of a treatment that made headlines over the past 2 years ago, past 2 years ago, and it is the mRNA vaccine for colon cancer. The mRNA vaccine may seem familiar as it is the vaccine that is used for COVID. So, what is the exact blueprint of how this vaccine works? So first, the you have to start with the design of the mRNA, and it begins with the DNA template and the in vitro transcription. And this template includes the open reading frame, which is the region that includes the protein coding segments and encodes the target antigen, along with the five translated regions and the three untranslated, the five prime untranslated regions and the three prime untranslated regions, which are crucial for mRNA stability and translation efficiency. So basically, the translation process for mRNA is very crucial. So basically, this is how the translation process goes. Capping, capping. So then it goes to the newly synthesized mRNA receives a 5' cap and a poly A tail at the 3' end. See the diagram here. And then it is translated. It begins the translation initiation, and the 5' cap protects the 5' cap protects it from the mRNA from degradation, and while the poly A tail enhances stability and translation efficiency. And finally, the mRNA is purified to remove impurities, such as truncated or degraded products, ensuring that only high quality mRNA is used for vaccination. Uh, so after the mRNA is translated, it goes to the cell, which it is translated by this weird looking thing, which is called lipid nanoparticles. And they are very small spherical particles composed of lipids, and they protect the mRNA from degradation and facilitate cellular uptake. And then the cell uh, tell will tell, and then the mRNA will tell the cell what to do. So once injected, the mRNA is translated into the target host cells, into the target protein inside the host cells. This protein is processed and presented on the surface of the antigen presenting cells. And the presence of the protein triggers an immune response in the innate immune response, in which it recognized the mRNA as foreign and activating the pattern recognition receptors, thus helping to activate immune cells, and also adaptive immune response, in which the antigens are presented to T cells, which I will further explain uh, uh, later, leading to the activation of helper T cells and cytotoxic T cells, which target and destroy cancer cells. So what innate immunity means is that it is in all animals. It is the recognition of traits shared by broad ranges of pathogens using a small set of receptors, and it usually has a very rapid response. So this is like your first response. And this includes like skin or mucous membrane secretions. And it also includes phytocytic cells, natural killer cells, antimicrobial proteins, and the inflammatory response in your body. And adaptive immunity only includes vertebrates, which this is like the second responder to an infection or any kind of disease. And it is the recognition of traits specific to a particular pathogen using a vast array of receptors such as cytotoxic cells or T cells or antibodies to defend against infection and body fluids, and it has a much slower response compared to the innate immunity here. So onto T cells. So T cells cannot directly bind to the pathogen or do not directly interact with the pathogen. But the T cells bind to antigen fragments displayed or presented on the host cell. And these antigen fragments are bound to the cell surface, proteins called histocompatibility complex molecules. And these molecules are host proteins that display the antigen fragments on the cell. This is here. And they are really important because they can go and kill the cancer cells. So there are certain risks attached to this experimental treatment, such as the risk of autoimmune response, especially if you are autoimmune compromised, and allergic reaction. The effectiveness will vary because everyone has a different uh composition of health or lifestyle choices. So we're not really sure how each person will react. And here are the references. All right, that will be all for my video today. Thank you for watching. Have a great day.